Okay, good morning, all of you. In today's class, we'll discuss about the other gene transfer methods. Yesterday, also, we have discussed about the transformation methods in bacteria, wherein we have discussed about calcium chloride method and electroporation. These are the two important methods used for bacterial transformation. In addition, what are the other gene transfer methods which can be used for bacteria as well as other eukaryotic cells? It could be yeast or plant cells or any other mammalian cells. So now let's discuss about the other gene transfer methods, which means other than calcium chloride method and electroporation. So the next gene transfer method what we have and which is very effective is particle bombardment gun. Definitely this method is not new for you. All these gene transfer methods I have already discussed while discussing about the steps involved in the production of recombinant DNA molecule. But here we are discussing all these methods once again in detail under the head of gene transfer method. So this particle bombardment gun, this is the technique that involves the bombardment of cells with high velocity microprojectiles. So what happens here is this gene gun, this is also called as gene gun. It shoots the foreign DNA into plant cells or the tissue at a very high speed. That's why it is called gene gun. It is also known as, the other names for this technique are particle bombardment, particle gun method. As I told you, it is also referred to as gene gun, ballistic process, microprojectile bombardment, or it is also called as particle acceleration. So basically it shoots the foreign DNA into the target cell at a very high velocity. So this is the apparatus of gene gun or particle bombardment gun. Basically, it contains a chamber in which the helium gas is flown and there is a rupture disc which is placed very close to the microcarrier assembly. This is microcarrier assembly made up of plastic. There is a rupture disc which is placed very close to this plastic microcarrier assembly, and on this microcarrier assembly we have the tungsten particles that are coated with DNA. These DNA coated tungsten particles are called micropellets, and these micropellets or microprojectiles, they are shot at very high velocity by the helium gas, and when they are shot at very high velocity they deliver the foreign DNA into the target cell. And here you also see a screen called stopping screen which is placed between your microcarrier assembly and the target cells, and these are the target cells that need to be transformed. So between these two things, which two things, microcarrier assembly and the target cell, there is a stopping screen, and this stopping screen can allow the passage of these microprojectiles, and then when they are when they are hit, when they hit the target cells, the DNA coated on the micropellets or microprojectiles is delivered into the target cells. So these target cells are transformed, and these transformed cells can be then regenerated on appropriate nutrient medium. So what are the components of this assembly, the particle bombardment gun? There is a chamber, and this is connected to outlet so that a vacuum can be created. Helium gas is flown inside the chamber. There is a rupture disc and microcarrier assembly on which the microprojectiles. What are these microprojectiles? The tungsten particles coated with DNA, they are present on this microcarrier assembly, and there is also stopping screen between this microcarrier assembly and the target cells, and it allows the passage of these microprojectiles through the stopping screen, and it allows the these microprojectiles to hit the target cells that are to be transformed. And this stopping screen is to control the speed of these micro projectiles before the DNA is delivered into the target cells. So it is important to draw this particle bombardment gun apparatus and name all the different parts of this apparatus when this gene transfer method is asked separately. General, it has been asked many times for 5 marks. So whatever the details we have discussed about particle bombardment gun, as I told you, it consists of chamber which is connected to an outlet to create vacuum, and the helium gas flows in the cylinder. There is micro carrier which is placed close to the rupture disc, and this micro carrier contains the DNA coated with uh, contains DNA coated tungsten particle which are called microscopic pellets or microprojectiles, and there is stopping screen which is placed between the target cells and microcarrier assembly. This helium gas is flown in the cylinder at a high velocity. So the stopping screen allows the micropellets to pass through and deliver DNA into the target cells, and then the transformed cells are regenerated on appropriate nutrient medium. So this is how the particle bombardment gun works uh, for the gene transfer into the target cells. The next gene transfer method available is micro injection. As the name sounds, micro injection, here there is a glass micro pipette which is used for injecting the foreign DNA into the target cell. So this micro injection is the technique of delivering foreign DNA into living cell by using a micro pipette, and one end of this glass micro pipette, it is heated. One end of this glass micro pipette is heated until the glass becomes liquefied. You all know glass, when it is heated to the appropriate temperature, it gets liquefied, and don't allow it to completely melt. You just heat one end of the glass micro pipette in such a way that it becomes slightly liquefied. When it becomes slightly liquefied, what you need to do, quickly you stretch that heated end so that a very fine tip is formed at the end. So what are you doing here? A glass micropipette is used in this micro injection technique. One end of this micropipette is heated. When it becomes slightly liquefied, quickly you stretch it so that it forms a very fine tip at the heated end. And this tip, the dimension should be about 0.5 millimeter diameter, which resembles a injection needle. So this glass micropipette which is used in this micro injection, you mend it in such a way that the tip resembles the injection needle. It should be so fine, should contain about 0.5 millimeter diameter. So look at the apparatus of this micro injection. So this is the micro injection needle which is used for delivering foreign DNA into the target cell. So here the cell which is to be transformed or which is to be micro injected, those cells are taken in a container. Usually it could be petri dish, and we also use holding pipette. This holding pipette is used for gently holding the target cell that is to be transformed, or here to be precise, to be micro injected. So using the tip of this holding pipette, gently hold the target cell that is to be micro injected, and you take your micro pipette, which is the glass pipette and which is already mended according to the need. So it resembles the injection needle, and allow that needle to pass through the cell membrane and deliver the contents in the cytoplasm or the nucleus of the cell, and then take out the empty needle out of the cell. I hope you are able to follow. And one more important thing is that this whole process of micro injection is carried out under microscope. You take the container of the cells in the microscopic field and use a holding pipette for gently holding the target cell that is to be micro injected. Then take your glass micro pipette and let it pass through the cell membrane of the target cell and deliver the contents inside the cell, whether it is cytoplasm or nucleus, by viewing under the microscope, and take out the empty glass pipette out of the cell. So the foreign DNA which is to be delivered inside the cell is delivered by this process of microinjection, and as I told you, the whole process is carried out under microscope. So this is microinjection. Once again, quickly recollecting the points, this is the process of delivering foreign DNA. This is done under a powerful microscope. Cells to be injected are placed in a container, and we also use a holding pipette. It is placed in the field of microscope. The holding pipette holds the target cell at the tip. When gently sucked, the tip of the micropipette is injected through the cell membrane. Then the contents of the needle are delivered into the cytoplasm, and then the empty needle will be taken out. That's how the whole protocol of microinjection. Then let us also see what are the methods we can use for transforming the yeast cells. As we all know, these yeast cells are taken as eukaryotic model system because of several reasons. Because they exhibit a very fast growth, and they also have dispersed cells. In addition, these yeast cells also have well-defined genetic system and versatile DNA transformation system. That's the reason for studying this phenomenon of transformation, or which method works better. We for eukaryotic cells, we take the yeast organism as the model organism, and it can be used as a effective system for protein production too. So most of the species of this yeast, including your very common species Saccharomyces cerevisiae, many of these yeast species, they may be transformed by the exogenous DNA in the environment. They may be transformed by the exogenous DNA in the environment, for which the yeast cells are treated with enzymes so that their cell walls are degraded, and then they eat spheroplasts, and then the cells are very fragile, but they take up the foreign DNA at a very high rate. So to transform these yeast cells, first we need to treat them with the enzymes that degrade the cell walls, and then allow the transformation to take place so that they take up the foreign DNA at a high speed, relatively in an efficient manner. So the recombinant DNA, for the recombinant DNA technology in yeast, a number of vectors had been constructed. We have studied so many vectors that could be used for yeast organism. Just to name few, we have yeast artificial chromosome and yeast episomal plasmid. These are all the different vectors we have studied, which are meant for transfer of genes in yeast cells. So what are the methods of gene transfer that can be used for transforming the yeast cells? There are several methods. Some of the common methods used in the transformation of these yeast cells are electroporation. We are not going to discuss again what is electroporation, how it works in yeast and all. Just we are naming it here. So suppose in the exam, if the question comes like what are the methods of gene transfer for eukaryotic cells, all these methods can be explained in detail. So the methods used for yeast transformation are electroporation and particle bombardment gun, that means ballistic method, and also glass bead method. So these are the methods used for transformation of yeast cells. So whatever the methods mentioned for yeast cells, these are commonly used for Saccharomyces cerevisiae, but they can also be used for transforming the other yeast species like Schizosaccharomyces fumbae, or it could be for other fungus like Candida albicans, and also can be used for Pichia pastoris. So these are all the different yeast and fungal species for which the mentioned transformation methods can be used, and it also includes other filamentous fungi like your Aspergillus species. So these are the transformation methods used for eukaryotic cells or specifically the yeast cells. So these are the gene transfer methods. After we discussed about bacterial transformation, that included the methods of calcium chloride and calcium chloride treatment and electroporation. The other gene transfer methods that we discussed today are particle bombardment gun, and also we discussed about micro injection. As I told you, particle bombardment gun method, basically it shoots the foreign DNA with a very high velocity to the target cells. We discussed about the apparatus, and then target cells, the cells whichever transformed will be grown on a grown on an appropriate nutrient medium. Then coming to the micro injection, the whole process is carried out under microscopic field. We use a glass micropipette, one end of which will be heated and stretched to form very fine tip, and then a holding pipette is used for gently holding the target cell to be transformed. Then with the fine tip of micro injection needle, the contents of the DNA will be passed through the cell membrane, and it will be gently delivered into the cytoplasm of the cell, and the empty needle can be taken out. So these are the techniques we discussed, and at the end we also discussed what are the techniques that can be used for transformation of yeast cells. The techniques mentioned are electroporation, bioelastic method, and also glass beads method. So this is all I wanted to discuss in today's class. If you have any questions, you can ask me. Any questions from your side?